Good morning, everybody, and thanks very much for the invitation to speak. What I wanted to do is talk a little bit about the approach to precision medicine in cystic fibrosis that Vertex has taken forward. And obviously my disclosure is I do work for Vertex, and so bear that in mind as you hear what I talk about. But I also wanted to pick up one of the themes earlier that was talked about the UK trying to be a leader in this area. And Vertex is a relatively small company. I suspect most people haven't heard of it. Boston-based biotech, but we've been also have a base in the UK at Abingdon, where we do a lot of research. And really 250 employees. We have Milton Park. We have a base in Paddington as well, where our international team is focused. And indeed, most of our intellectual property is also based in the UK. And this is just an example of that commitment to doing this kind of work in the UK, because we recognise this is a very important area, and important, particularly in the Brexit times, how we go forward. So we're currently doing 14 active trials in the UK, another 6 in Ireland, and interestingly, the next generation of molecules, which I'll talk about at the end, which have some very exciting potential, is being exclusively done in the UK for one of these molecules. So we do have a real strong commitment to this, as well as other trials and to investigator-initiated studies in the UK. So I hope we could be part of that process of helping UK drive forward. For my talk, what I wanted to talk about is the vision that we have, which is to deliver really high levels of benefit to CF patients as early as possible, to really try and transform the trajectory of the disease for the patients. And the framework I've adopted is to talk a little bit about the precision medicine approach based on the genotype, talk about the potentiators and correctors, the ways in which we can really tailor the treatment to the genotype, talk about some of the future molecules which enhance that, and then talk a little bit about the way that's really demonstrating outcomes, both acutely, but also that long term. Part of that question about, in diabetes, is it cardiovascular risk? Well, in CF, are we beginning to be able to see a difference of really altering the outcome for the patients? And where we start a little is with the genotype and phenotype. We've heard this mentioned several times already today. There's an excellent database in CF. We're incredibly fortunate that some very far-sighted people 20 years ago started setting up really robust registries. The US registry captures over 90% of all the CF patients. The UK registry has every UK CF patient in it. So we have fantastic data on the patients and on their phenotype. So we can look at their age, their genotype, their gender, sweat chloride levels, etc., nutritional parameters, that gives us a really strong handle on both the genotype and the phenotype. There's a great progress now on understanding the actual molecular defect caused by each of the variants in the Cftr transmembrane receptor gene. So we can begin to look at each genotype, what that does functionally, and then start to then design and develop modulator therapies to address those specific molecular defects. And where that's now backed up is we have experience from over 60 clinical trials with these modulators, so we can begin to see not just the theory, but that translated into clinical practice and into clinical outcomes. And this was really driven initially by, way back in 1989, the gene was discovered and identified. And of course, there was huge excitement at the time that we could cure the disease. And here we are, 30 odd years on, we're just beginning to make inroads. So I think there is a time course for this. But what we've really understood much better, I think, is the dysfunction and the phenotype. Normal person, has a 100% Cftr function as measured by sweat chloride. That's our most typical measure of the function of the Cftr protein. Carriers, around about 80%, but they're completely normal. But as we track back, there's CF-related diseases. So people who carry mutations associated with some impairment, not enough to cause CF, but they have somewhere around this region of Cftr function, but modest expression of disease. Most typically, congenital bilateral absence of vas deferens might be an example of a related disease. There's a group of patients much better identified now with genotyping, of course, with a delayed onset, a less rapidly progressive form of the disease, where they'll have perhaps somewhere in the range 10, 20, 30% of normal function. And then the patients with the classic severe CF that we probably all think of from the textbooks, with virtually no Cftr function. But what this tells us is, of course, the more we can increase the Cftr function, the better the long-term outcome should be. And one of the approved molecules, the combination of lumacaftor and ivacaftor, is able to produce something of the region of 15% of normal function in these very severe patients. Good step forward. It moves them perhaps in this direction from being severe to being in a less rapidly progressive form of disease. But the first molecule that was approved, the ivacaftor molecule, in this particular mutation, G551D, you can see we got remarkable correction, up to 60% of function. So in a sense, you're taking someone who is a severe patient and potentially putting them into this category where they have a very different form of the disease. And the way this works is in a normal person, CFTR protein is an epithelial surface protein that transports chloride primarily, bicarbonate as well, but primarily chloride transport, and that's why we measure sweat chloride in the skin, because of the defect in that protein. And the way we tend to think about this conceptually is the molecular defect in the CFTR mutation are now well understood, and they either cause a decrease in function or a decrease in quantity or both. And so, for example, you can have mutations in class 3, class 4, where the protein is at the cell surface, just doesn't function. And if we could drive that function, we'd get benefit. The most common mutation is the delta F508 mutation, where there's really very little protein produced. And even when it does get to the surface, it doesn't work very well. So you have a problem of primarily decreased quantity and a lack of function. And that's really how we set about trying to solve the problem. So we know now that there's 250 known CF-causing mutations identified. Again, from this database, we've really been able to look at, there's 2,000 odd mutations identified, 250 are clearly disease-causing, and they're being characterized. So there's a process going on to characterize every one of those 250 to understand exactly what the molecular defect is. Many of them have decreased function. So the gating mutations, the protein doesn't open and close properly, or they have some residual function. There's protein at the surface, it doesn't conduct so well, it doesn't open so well, but not to such a severe degree. So that's one categorization of function. The very common F508del, the delta F508 mutation, many patients, about 40% of the patients in the world are homozygous. They have a decreased quantity due to defective processing and trafficking of the protein to the cell surface, so it just doesn't get there. There's another group of patients who have the 508 on one side, minimal function mutation on the other, and they have a decreased quantity, but the other allele is essentially non-responsive to the currently available treatments. And then the last group of functional defects is patients who just don't produce the protein. The class 1 patients, they produce truncated protein that never gets there. So how did we set about trying to then tailor treatments to these defects? And working from the top down, the biological problem at the top is defective function. So there's protein at the cell surface, not opening and closing, not working. And for those, we use a class called potentiators, of which ivacaftor is the first example. That's been approved now for 5 years to treat patients with very specific set of mutations. It's probably around about 5% of all the CF patients. So it's just 5% of an incredibly rare population already, but making a dramatic difference. In a way, the trickier problem is this group of patients, the class 2 defects typically, where the processing and trafficking, the protein doesn't fold properly, doesn't get to the cell surface, it gets degraded. And for that, we developed, so far, a number of correctors. And you might be surprised by the list of correctors. Lumacaftor, in combination with ivacaftor, is approved. Tezacaftor is the next molecule in the pipeline being developed. That, actually, data will be released next week, quite probably publicly. But there's a series of molecules behind that because we're trying to make sure that we do better and better and better. So one goal is to provide a treatment. The next goal is to provide a better treatment for everybody. But for the patients, perhaps with the class 1 mutations where they have a defective protein synthesis, it's not even getting out to then get folded, we need different approaches. So we're looking at gene editing approaches with CRISPR or mRNA replacement, or the UK Gene Therapy Consortium has also been making great strides down that path. But that's fundamentally how we're going from understanding the genome to the protein defect to then the treatment options in precision medicine. And that's really what this slide is intended to illustrate, that we have really good data now on a clinical phenotype. So we can understand somebody's age, their BMI as a measure of nutrition, their pancreatic status, are they pancreatic sufficient or pancreatic insufficient? What's their lung function like? Are they 50% predicted typically or 90% predicted typically? Where's their sweat chloride? You know, severe CF would be 100, Normal would be 20 or 30. 80 might be typical for one of the residual function mutations. But we can understand really well the phenotype. We can associate that with then the understanding of the molecular defect. Is the protein at the cell surface? Is it opening and closing? Is it a folding defect, etc.? And then we can combine that with experimental evidence, both in vitro or ex vitro, about a predicted clinical response. But then we need to do the clinical studies to evaluate that, to confirm and inform that combination. And so let me talk a little bit about the combination of these molecules and how we do that. So far, this is the group of patients that have treatments available. They're approved for use, not necessarily reimbursed, but they're approved for use. So for Cftr, it's approved for use in children 2 and above with gating mutations. They're a collection of 10 rare mutations. And in fact, there's trials now going down as close as we could get to births, because we want to have the ability to stop the disease in its tracks. And if we can start when they're 1, when they're 6 months, when they're 3 months, we may be able to make a dramatic difference to the long-term trajectory. The R117H mutation is approved for subjects 18 years and above because of the way the clinical trial data worked out. The lumacaftor-iva combination is approved for subjects aged 12 and above in the US and in Europe for patients homozygous for the delta F508 mutation. But the problem is, how are we going to help the rest of these patients? Because the ambition is not to leave any CF patient behind. We want to help everybody. And so to do that, we're taking an example, perhaps, of residual function CFTR. So these are the more moderate defects in the amount or function of protein. They're still associated with disease, and they're still associated with, typically, premature mortality. Patients with these mutations die maybe 10 years later, so on average, late 30s, early 40s. So still far too young, but less severe than other CF cases. We have clinical evidence that they have functional CFTR at the cell surface. So their sweat chloride might be 70 or 80 rather than 100. They'd typically be pancreatic sufficient, not needing pancreatic enzymes. The lung function decline would be less rapid. So we know they have some evidence that they have some functional CFTR. We can look at the nature of that defect and then look in vitro at what that response is and the response to treatment. So we've looked at human bronchial epithelial cells. From patients having transplant and things, we can get human bronchial epithelial cells, test them in vitro to look at responses. We can make fish or rat thyroid cell constructs, put the gene defect into those cells, measure the response. There's another program really being championed out of the Netherlands where you can grow gut organoids. They take rectal biopsy tissue, grow those into little spheres of cells, and then look at their response, their swelling to treatment. Or intestinal current measurement. But again, we still need to inform that with clinical trials to see if we can translate the in vitro understanding to clinical understanding for the more common mutations. There is a limitation when there's only 10, 12 in the world with a certain mutation. You can't do clinical trials, and we have to crack that problem differently. But what we have been able to show is that there's a good correlation, a good relationship between measurements of in vitro chloride transport. So we're measuring the amount of chloride transport as a percentage of the wild type, as a percentage of normal, and going that way, the more CFTR function, the better. And comparing that to sweat chloride, and the sweat chloride changes. And again, the lower the sweat chloride, the better the function, the better the patient. And we see a nice relationship, and the open cells being pre-treatment, post-treatment. But there's a good relationship that says what we see in the in vitro models will predict what we see in vivo in terms of sweat chloride. So how do we then take this forward in a real kind of precision way for the patients? And the goal really is to think about treating both alleles. To have CF, you have to have a defect in CFTR gene on both alleles. The vast majority, 90% of patients, will have a 508 del mutation on one allele. And then on the other side, we'll have potentially a minimal function mutation, typically class 1, something we can't correct. And what we would expect is a fairly minimal response, because in effect, we can only make a difference to that mutation. For this group, the delta F508 homozygous, others correctable, we can get a benefit from treating this mutation and that mutation. We would expect some more gain. But for the patients with gating and residual function mutations, because of the potency and the effect of Cftr, we would expect much more clinical gain. So the picture of what we would expect for patients really varies by their precise makeup of the genes on two alleles. And how does this translate in vitro? We have data for the G551D patients. That was the mutation for which ivacaftor was originally approved, in combination with 508. And you can see, well, hopefully it projects okay at the back. So patients receiving neither ivacaftor, cell systems with neither ivacaftor or tezacaftor, really no chloride transport. Add ivacaftor, you get a very dramatic response, which is why we saw the very dramatic clinical picture. But what we could see is when we were also using tezacaftor to get the 508 protein to the cell surface and then make it work more with ivacaftor, you get more response. So we can make the patients better. And so that's led to clinical studies of tezacaftor in patients with the G551D mutation plus F508 and an additional range of studies. Those studies haven't read out yet, but our goal is to see if we can, by treating both alleles rather than one, generate additional clinical benefit. In a similar way, we looked at this R1070W mutation. That's associated with a decreased amount of protein. You can see ivacaftor, tezacaftor, very little function. Add ivacaftor, you can make the protein that's there work a bit harder, but if you had tezacaftor to help get that protein to the cell surface and then stimulate with ivacaftor, much more response. And alternatively, you have mutation like the R352Q, decreased function, some underlying residual function of Cftr. Add ivacaftor, you drive that more. Add tezacaftor, you drive it a little bit more. So there's a study of these 25 residual function mutations that will be reported next week to try to see if we can translate the science of being able to treat these patients into clinical benefit, because that's really the goal behind all of this, is to make people better. A further step is then in the next generation of molecules and what we want to do to try and continue to enhance the benefit. And the real example that we use is maximizing the amount of function of the 508, because that's 90% of all the patients in the world. If we can make a big difference on that one allele, just this single 508 allele, then we can help 90% of the CF patients much better, even if the other mutation is non-responsive. So that's the goal of our next generation, the next program that we do. And this is an example of how we've been trying to take that forward. So this is the in vitro system looking at a 508del minimal function human bronchial epithelial cell system. And back in 2013, to give context to this, I should say the Teza-IVA, the 661-IVA, we're getting about this level of correction in a cell like that. So, something of the order of 10-15%. We got what we call a hit, a molecule that seemed to have a function that we thought could be beneficial. I've been working on optimizing that through 2014, and you can see we're looking to increase the potency, but then there was an enormous amount of effort in chemistry and biology to look at, as you can see, a very large number of molecules, really looking to find molecules up at this end of the spectrum. Ideally, we'd get to carrier levels of CFTR function, so around 80%. So, like, I might be carrier, you might be a carrier, but you don't know, you're normal. And if we can get drugs that will get patients up to this level of correction, and there are a few examples, then we could hope for dramatic benefit for the patients. And this is an example of how that in vitro data is working out. So this is all on a background of the tezacaftor-ivacaftor combination throughout, other than the base, and then adding one of these three molecules. These are what we call a next generation molecule. They work on a different part of the protein folding so that we have a complementary effect. And I think it's clear with the 152, 440 and the 659, we can get much stronger sweat chloride response as a potential of function. And this, to remind people, G551D, we got this level of correction. So we should be able to get that level of benefit for any patient with a single delta F508 mutation. And what that would mean is that there's still the patients that don't have delta F508 that have mutations responsive to ivacaftor, either H2 or 18, depending on the particular mutation. But in this big block then, for the 508 homozygous patients, the combination of the first and second generation should give them enhanced benefit, so we'll get much better clinical benefit for that population. And for the population with minimal function, we can expand a benefit to that population that has nothing, which of course leaves us with then roughly 10% of the CF patients who currently have nothing. And that nothing is really then addressed by replacing or fixing CFTR with mRNA, with CRISPR technologies in the future. So that's the approach we're trying to really tailor what the mutational defect is to the phenotype, to then choice of clinical treatments. And the key question is then, how is that working out in clinical practice? And there's a couple of components to this. I showed you this already. The in vitro chloride transport correlates well with sweat chloride. So we know our in vitro systems are likely to be very predictive of a clinical response in terms of sweat chloride in vivo. So for mutations, for example, where you can't do clinical trials, there just aren't enough patients, if we can show in vitro a response, you would expect a sweat chloride response. And then, of course, the question is the so what? It's like reducing HbA1c. Does that translate into clinical benefit? Well, improving sweat chloride, does that lead to clinical benefit? And the answer is at a population level, yes. This is an analysis we did of sweat chloride, the absolute change in sweat chloride, where the improvement in sweat chloride, the lower your sweat chloride, the bigger it goes down, the more benefit you get. And we looked at the relationship with that, an absolute change in lung function as measured by percent predicted FEV1. And at a population level, you can see there's a really reasonably good correlation that if you can show that you improve sweat chloride at a population level, you would expect a population level benefit in lung function. Now, it really breaks down at an individual level because there's so many other factors going on in infection and modifier genes and a host of other factors. But at a population level, it's strongly predictive. And so how's that translated into clinical practice? Ivacaftor, the first approved molecule, increased lung function by 10%, halved exacerbations, improved nutrition, reduced symptoms dramatically. I mean, it's helped patients who were on a transplant list come off the transplant list. It's been transformational for these patients. What we did, though, is looked at a range of things in terms of disease progression. And I'll finish in a couple of ticks. So ivacaftor, you get this big spike in lung function. But then we looked at the rate of decline across time on ivacaftor and compared it to controls. And because 90% of the patients who are eligible for treatment with ivacaftor are on it, we can't compare untreated controls. So we took a control population. But you can see the rate of decline in lung function was approximately halved. It was 47% lower. So we're changing the long-term trajectory of the decline in lung function for the patients. In the interest of time, I won't deal with, there are limitations with using registry data and matched controls and so forth, so we recognize that, but still in a rare population, registry data is probably the best way to address this topic. But ivacaftor long-term has statistically significantly reduced deaths at years 2, 3, and 4 of treatment. I mean, you're 60% less likely to die on ivacaftor. Transplantations reduced, hospitalizations reduced dramatically, exacerbations are reduced dramatically. So the promise of the sort of precision medicine is translating into real-world clinical benefit for the patients. What's intriguing, though, and we're just starting to understand this, is that the decline in FEV1 and the impact on rate of decline is not correlated to the acute change. So there's probably two processes about what we see acutely in lung function and what you see in terms of the long-term trajectory of the disease, which is why with the lumacaftor-ivacaftor combination, the lung function improvement was smaller, but the decrease in the rate of decline on lumacaftor-ivacaftor compared to the controls was 42% lower. So it reflects that picture of taking someone from a very severe to a less rapidly progressive disease. And so really to wrap up, I think where we've started is we have the genetics, we've identified the root cause of CF, the Cftr gene defect. We've got really good data on the correlation between the level of dysfunction and the disease phenotype. We've designed medicines that really try to target the specific genetic defect, and now we've demonstrated both acutely and in longer-term data that that is changing the disease. And that's our target, to carry on doing that. This is the portfolio of treatments, and you can see there's a huge pipeline of effort going on behind it with the 2 approved molecules to make sure that we can really provide something fantastic for everybody in CF. And with that, I'll say thank you and take any questions.